Thank you, Tia, and thank you very much to Jill for organizing this symposium today. I'm going to switch gears a little bit and I'm going to speak to you about our findings from a behavioral phenotypes that we find in a mouse model of paternal saccharin and nicotine exposure. And so, as everybody is aware, nicotine is a highly addictive substance and it is found in a number of tobacco products. These products include traditional cigarettes, smokeless tobacco, and electronic cigarettes. And anyone that has ever smoked any of these substances will agree that cigarettes or tobacco is one of the most difficult substances to actually quit. More recently, e-cigarettes have been marketed as being safer or a safer alternative to regular cigarettes. And this is because they're believed to not have the harmful ingredients that are contained in cigarette smoke. However, e-cigarettes or vape products do indeed have their own issues. These are extremely appealing to younger population. They are available in a variety of flavors, and many of them contain sweeteners, which is why I am speaking about them today. In fact, if you look at smokeless tobacco products, whether it be e-cigarettes or smokeless tobacco, they actually contain saccharin at levels nearly 25-fold higher than in food products. So most of you, and I'm sure like me, would have thought of saccharin. We think about it in food. We don't necessarily think about it in smokeless tobacco or e-cigarettes. And saccharin is traditionally used as an artificial sweetener. You know, it's a sugar substitute, and you see it in substances that have labels of diet or light. About 40% of people in the United States, or adults in the United States, actually consume low-calorie sweeteners such as saccharin. And low-calorie sweeteners are deemed to be regarded as safe for human consumption. But some of these substances, including saccharin, can actually activate reward circuitry in the brain. And so it may actually have a neurobehavioral phenotypes that could be present. And this is something that we are interested in the lab. So now there's a legitimate concern for exposure to saccharin alone through food and saccharin being co-exposed with nicotine through the use of e-cigarettes and smokeless tobacco. And we can actually mimic this quite well in the lab. And what we do is we use a mouse model. Typically, when mice are exposed to nicotine, one of the ways to do this is to do an oral exposure. And in order to administer the nicotine, we actually use sweetener, which is saccharin, to basically mask the bitter taste of the nicotine to make sure that the mice will actually ingest the nicotine water. And so we take these male C57 Black 6 mice and we expose them to either plain drinking water containing 2% saccharin, or drinking water with saccharin plus nicotine. And the nicotine that we're exposing them to is roughly the equivalent of a packet of cigarettes per day. And so these mice are exposed to the 3 different types of water bottles, and then we breed them with drug-naive females to produce the F1 generation. And then these F1 generation are put through a battery of behavioral tests. And basically, the tests that I'm going to speak about today are tests that we performed that are associated with dopamine transmission in the brain. So we've looked at spontaneous locomotor activity, spatial working memory, object-based attention, and locomotor or motor-impulsivity, excuse me. And we basically test these in the F1 offspring. The goal here was to examine how many of the phenotypes, or if any of these phenotypes, are indeed inherited. So in order to fully understand the inherited phenotypes, it's important to ask a number of questions. And one of the questions that we asked is, what are the factors that would influence heritability of the phenotype from one generation to the next? And today I will introduce some of the factors which played a role in our mouse model. The first thing that we needed to establish was how long are we going to expose these male mice to either nicotine plus saccharin or saccharin in their drinking water? At what time point do we take this male mouse, breed him with a drug-naive female to produce the offspring, and then put them through a number of behavior tests? We basically started out with an 8-week exposure. And we chose this 8-week exposure because we wanted to make sure that we encompassed a full cycle of spermatogenesis, if not a second one. And so since spermatogenesis is believed to be, the cycle of spermatogenesis is believed to be 5 weeks in a mouse, we began with our 8-week exposure. And we found that 8 weeks of exposure of the male mice to the respective drinking water did not produce any behavioral phenotypes in the offspring, and neither did the 10 weeks. And thankfully, we persisted, and 12 weeks was the sweet spot. And we did actually find heritable phenotypes in the offspring, which was a relief because there's only so much negative data one individual can handle. And so, having established the time needed to produce heritable phenotypes, we next focused on the sex of the offspring. We asked the question, does the sex of the offspring influence heritability? And what I'm showing you here is a graph that represents spontaneous locomotor activity. We test the spontaneous locomotor activity over a 12-hour period when the lights are off. And this is typically when a mouse is more active. And what you can see is that our co-exposure to nicotine and saccharin is our purple line. Exposure to saccharin alone is our blue line. And both of these treatment groups actually produced a heritable phenotype in the offspring. Both the saccharin and nicotine plus saccharin-exposed fathers produce hyperactive F1 male mice. When we look at the female mice, it's actually a little bit different. So we do not actually detect a significant difference in the female mice. And this indicates that there's a sex-specific transmission of this locomotor hyperactivity phenotype. In addition to locomotor activity, we also looked at some cognitive tests. We examined spatial working memory and motor impulsivity. And basically what I'm showing is the same thing here. It is the male mice and not the female mice that appear to have inherited the phenotype. And so this data, this behavioral data, led us to the next factor. And although we set out to examine the effects of nicotine exposure, we quickly realized that the substance we use to mask the bitter taste of the nicotine also produces heritable phenotypes in the offspring. What you can see here from this graph is that co-exposure to nicotine plus saccharin in the purple have spatial working memory deficit in the F1 male mice. However, motor impulsivity is impaired in F1 mice produced from a saccharin-exposed father. And so this brings us to the father. Since the F0 is the exposed individual, they're the generation that are exposed to either the saccharin or the nicotine plus saccharin, we then ask the question, what information can we gather from this father, which may actually predict the phenotypes inherited in this F1 offspring? And so there's a number of ways that we can do this. And we decided to first look at the somatic cells, and we use behavior as our output. And we ran the male mice through the same behavioral test that the offspring were run through. And what we found was that the father, a saccharin-exposed father, and its son had impaired motor impulsivity, showing that they actually shared one of these phenotypes. However, if you recall from earlier, the hyperactivity phenotype is unique to the F1 male mice, and the female mice have no behavioral phenotypes at all. And so here you can say that in some ways the father does predict, but it's not a great prediction. The story for the nicotine plus saccharin exposed fathers, absolutely no behavioral phenotypes whatsoever detected in these mice. However, the son, if you recall, has working memory deficit and displays a hyperactivity phenotype. So the F0 behavior itself did not necessarily predict the F1 phenotype that we would see. And so the other alternative, if you're studying the father, is to look at his germ cells and look at epigenetic modifications. And so we focused on DNA methylation as our epigenetic modification. We did this using a double swim assay, isolated the mature sperm, extracted the DNA, and looked at 5-methyl cytosine content in spermatazoa following 12-week exposure to either water, saccharin, or nicotine, co-exposure to nicotine plus saccharin. And what you can see here is that the F0 saccharin-exposed male mice have a significant increase in 5-methyl cytosine content. We do not detect any changes in 5-methyl cytosine in the Nic plus saccharin group. And what this basically does is perhaps separate the mechanisms by which the phenotypes are being inherited by the offspring. And so in order to look a little bit closer at the changes in the Sac germline, we looked at dopamine receptors. As I had mentioned earlier, the dopamine is a neurotransmitter that's known to regulate the behaviors that are inherited by the offspring. And what we do see is an increase in 5-methyl cytosine in 3 of the 4 promoter regions of the dopamine receptors that we actually examined. And so we can conclude from all of this that there are many factors that can influence heritability of a phenotype in offspring. But what about subsequent generations? And so it's obviously extremely important to look at not just intergenerational, but look at a transgeneration, and does the sex of the founder actually impact the future generations? And so using a different paternal nicotine exposure mouse model, we looked at intergenerational and transgenerational transmission. And what we found was that the pattern of intergenerational and transgenerational transmission is indeed influenced by sex. So what you can see here is that both male and female F1 mice show a behavioral phenotype, but this is only passed on through the maternal line, and it's only the F2 male that show the phenotype. And so in conclusion, we learned quite a bit from this study. We learned that 12 weeks of exposure was required in our model to produce the heritable phenotype. We also learned that the behaviors that we see are predominantly seen in males. Co-exposure to saccharin plus nicotine, as well as exposure to saccharin alone, produces heritable phenotypes. And And we also learned that you can learn quite a bit from the father himself, but it doesn't always predict the outcome in the offspring. And finally, I would like to thank all of the people that were part of this study and that influenced the results, and of course, thank our funding sources. And with that, I'm happy to take questions.
Wonderful. Thank you so much, Dierdre. We have a question from Francesco. Have you looked at the global methylation status in the germ cells of males exposed for 10 weeks? If those changes are present there too, they may not have been related to the phenotypes in the F1s.
Yes, so that is not something that we have done. I think we may have actually spoken about this at a previous meeting before. And one of my colleagues actually in the lab, Sarah Jones, who's going to speak later, her project is actually going in that direction. So we now know that we need to look at sperm from the shorter exposure. Yeah, it's a great question.
Yeah, and then I had a question too. Well, I had several questions, but I think one that I'm most curious about is that you didn't see any phenotype changes until the mice were about 12 weeks old. Are you in your previous slides? Are you looking at were you looking for other receptors in those previous exposures to see if there were changes there, but they just weren't showing a phenotype until they got to 12 weeks? Does that make sense?
I think I might not have been completely clear. So the exposure to the father was 8 weeks, 10 weeks, or 12 weeks. But then
Yeah.
when we examined the offspring were adults. They were 60 to 90 day old.
Oh, okay.
Yeah.
Okay. All right. Well, perfect. Thank you so much. Thank you.